I'm going to mostly focus on the technology on metaproteomics and what we can use it for, and I'm going to go through, like, basically can give like bird's eye overviews of different types of studies just to highlight the different types of questions that we can address with this particular approach. Right, so first I'll explain metaproteomics in a few studies, and then I'll actually, more towards the end, I'm going to talk about some of the dietary microbiota interaction studies that we're currently working on. Okay, so yeah, just to give a very general overview of all the different things that we do in my lab, we have actually currently a very broad portfolio of things that we're doing. We study more efficient carbon fixation pathways that we discovered in some symbiotic microbes in the past. We work on the genome and metabolic evolution of beneficial symbionts. We also look at bacteriophages in complex host microbe systems. But then really, currently, a lot of our projects actually focus on the impact of the microbiome on plants, as well as on actually looking at the interactions between dietary components and the microbiota. So we have a lot of different research areas, and I'm not going to talk about all of them today, so if you have any questions about them, I'm happy to chat about that afterwards. So yeah, to get into metaproteomics, just a very brief overview of the history of metaproteomics. It's not a very long history so far. The term was first coined in 2004, and it was proposed as the term metaproteomics for the large-scale characterization of the entire protein complement of an environmental microbiota at a given point in time. So that's what it was originally conceived, to try to characterize all the thousands of different proteins that microbes use in these communities. And I always like to put up this graph. A lot of people use these publication graphs to demonstrate something. What I'm trying to demonstrate here was this graph that actually, like, we looked at the term metaproteomics in PubMed. You can see, it took a while. Not a lot of people started doing it initially, but in recent years, we have really seen a heavy lift-off, an exponential growth, and people actually starting to use metaproteomics. And that's for several reasons. So why would we actually care to do metaproteomics, right? Why would we care to characterize all the proteins that microbes express in communities or host microbe systems? Right? Because there's a lot of different methods that we can use to look at these uncultured microbes, right? A lot of these microbes are not culturable, so we have to actually rely on some kind of cultivation-independent methods, right? And there's different types of cultivation-independent methods that we can use to characterize microbes in these types of communities. So for example, in one of the most basic techniques is usually looking at something based on genomes. So for example, we have metagenomics, which allows us to try to sequence all the genomes of the microbes in the communities to get an overview of the functional potential of the microbes. We can move on to, for example, metatranscriptomics, where we would be looking at expression of these genes. But ultimately, what we all care about is what actually happens in terms of phenotypes, right? We really want to know what's going on, what are these microbes doing, how do they interact, how do they function. And so ultimately, we want to look at the phenotype of the microbes at the molecular level in these complex communities. And for that, we have to either look at the protein level or we have to look at the metabolites, right? So we do metaproteomics or metabolomics to do that. One thing I like to highlight here is that, of course, it's relatively easy to link proteins back to the source organism, right? Based on the sequence, we can link it back to the source organism. It's much harder when we do metabolomics to actually say where did the metabolite come from and who is consuming it, because there's no taxon-specific signature within a metabolite that we can use to link it back to the species of origin, so it's much more complicated. So proteins have that advantage that based on the sequence, we can link it back to the species of origin. So when you want to do metaproteomics, there's a lot of different workflows that are currently being used, and I'm just going to give a very generalized workflow right now. So a general metaproteomics workflow would, of course, start with sample collection. You would be collecting your favorite types of samples from whatever environment you're interested in, and then you have to do some kind of cell lysis procedure, right? Some kind of sonication, bead beating to disrupt the cells, to extract the protein. You get a dirty protein mixture, which we then use. We clean up the protein mixture, and ultimately we digest the proteins, all those thousands of proteins. We're using a specific enzyme to obtain a clean peptide mixture in the end. And that's then going to be the input for the next step. A question that sometimes comes up in this context is how much material do we actually need, especially if you work with environments where you sometimes have very little material that you can obtain. It can be critical. And we can by now work with very, very little starting material, right? We can work with as little as 0.5 milligram of wet weight cell material or less by now. So we need very little material to actually get started in metaproteomics. Okay, so you have your clean peptide mixture, and then the next step is to actually separate these peptides. So we use one-dimensional, two-dimensional liquid chromatography, where we separate these peptides over many hours to get, because we're dealing with tens of thousands, hundreds of thousands of different peptides. We inject them into a high-resolution mass spectrometer, and we collect the mass spectra of the intact peptide masses as well as spectra of the fragment masses of the peptides that we select. So we ultimately end up with like hundreds of thousands of these fragment mass spectra, and we use them for the actual identification step. So the input for the identification and quantification of the proteins is really, we have to have these fragmentation spectra and as well as a protein sequence database. So we have some kind of FASTA file with a lot of protein sequences that we ultimately want to match to. And it's most optimal if these sequences are closely derived or actually derived from the same sample. So oftentimes what we do when we do these types of approaches, we actually sequence shotgun metagenomes, assemble them, bin them, get the genomes out and predict all the protein sequences to then use that as a database. It depends a little on how well your sample is characterized. So ultimately our inputs for the identification is a protein sequence database and on all the spectra, and that we use in different types of computational tools to identify the peptides and ultimately identify and quantify all the proteins in the sample, or saying all the proteins, as many as we can, right? Because there's always low abundant species that we might not get much from. But by now we can usually get more than 10,000 proteins identified and quantified in a sample. Okay, so yeah, and one thing that we do a lot when developing these approaches is we work with fully controlled samples. And that has been a really critical tool in our hands, right? And initially when people started looking at microbial communities, at these very complex samples, they just used whatever newest sequencing method came out or whatever newest method came out, and they just immediately applied it to environmental samples, and then much later realized that they did not properly validate all the potential artifacts that you get when you have a very complex sample. So what we do now is we always work with fully controlled samples to develop and validate our methods before we move into actually dealing with true samples, so we actually can anticipate what might go wrong, right? So in one of the samples we, for example, use a mixture of 32 bacteria, archaea, eukaryotes, and viruses, which we have in pure culture. We mix them all together in different ratios, make different types of mixed communities, and then we use that for the methods development. And I think I have an example later of one of these studies we did with these communities. Another thing that we do is, of course, if you have a nice mixture, that's a good thing, but also we want to look at fully controlled samples within the context of the true samples that we are looking at. So since we do a lot of work in plants right now and in animals, we have actually obtained multiple systems where we have fully defined systems, right? So for example, we work on a switchgrass system where we have one fungus and one bacterium infecting it. We have a corn system where we can grow the corn not abiotically with a specific community of 7 known microbes of which we have the full genome information. We also work with Jeff Dengel at UNC Chapel Hill, who has this beautiful Arabidopsis system where they have up to 200 different microbes they can use to infect the Arabidopsis. And then we're very lucky at NC State that we also have a germ-free facility, so we have obtained multiple fully defined microbial communities that we can use to inoculate germ-free mice to do very controlled experiments with them. Okay, so now I'm going to go, as I said, briefly through a few studies just to highlight the different types of questions that we can address using metaproteomics, and some of, and you can think about how that would relate to your potential systems of study. So I guess one of the most classical things that metaproteomics was used for initially was to really just look at metabolism and physiology, looking at all the proteins that are abundant and what can we learn about what's actually going on, what are the microbes actually doing in terms of metabolism and physiology. And so the first example relates to that. And I'm going to talk about one of my favorite marine systems here. So yes, so this relates to Olavius algarvensis. That's a gutless marine worm that lives in these beautiful sandy sediments off the coast of Italy. And what is really critical to know about these sandy sediments is that they're extremely poor in any nutrient and energy sources. So, and as I said, the worm is gutless, doesn't have a digestive system, doesn't have a mouth, cannot ingest any food. So when you look at the cross-section of the worm, you don't see any digestive tube in the middle. Instead, it has a very thick layer of bacterial symbionts that live under its outer skin, and they are like stained in red and green here. So those bacteria provide all the nutrition for this particular animal. So there were already a lot of studies that had been done on this particular system, metagenomic studies and physiological studies, to really figure out what's going on in the symbiosis. How does it actually work? How do the symbionts provide nutrition in a very energy and nutrient depleted environment? So what researchers had found before we did the metaproteomics is that within in this particular symbiosis, we have multiple different species of symbionts. So we have sulfur oxidizing symbionts that are autotrophs, they fix carbon dioxide and provide organics. And then we have sulfur reducing symbionts inside that actually provide internal recycling of sulfur compounds. We have an extremely energy efficient system. But one of the questions that could never really be fully answered was the question of what is actually the external energy source that drives the whole system, because of course this internal cycling cannot go on forever without any external energy inputs. So we did a metaproteomic study following a metagenomic study to learn more about what are the symbionts actually doing. We looked at all the most abundant proteins that they express, and we learned a lot about the different pathways they're using, how they interact with the environment, what substrates they cycle, and all these things. But just looking at the one question now about what is a potential energy source for the system, one thing that we discovered that we thought was really surprising at the time is that some of the symbionts have abundant enzymes for the use of carbon monoxide, which is a very poisonous gas. They have an enzyme for the use of carbon monoxide as an energy source. So we were really surprised at the time because we had never considered that might play a role in this particular environment. So based on what metaproteomics told us here, it really said, this is an abundant enzyme, you really should be looking into that. We of course went back and then did more targeted approaches to actually look for sources of carbon monoxide in the sediments. So we did measure pore water concentrations in these sandy sediments, and what we actually found is that the pore water concentrations of carbon monoxide are much higher within the sediment as compared to a seawater control from the overlying seawater. And later on we actually went further and studied what kind of materials in the sands might actually produce the carbon monoxide. What we also did is we actually took the worms, we put them in an incubation bottle where we added labeled carbon dioxide to these bottles, isotopically labeled carbon dioxide, and then we used an imaging mass spectrometry method called nanosims to actually look at individual cells in terms of the isotope composition. And to these bottles we added carbon monoxide to see if carbon monoxide would increase carbon fixation. What we ultimately found is that indeed some of these symbionts, especially this gamma-3 symbiont here, increases carbon fixation when carbon monoxide is present as an energy source. So metaproteomics really guided us very well here to ultimately discover a completely new physiological trait in this particular symbiosis. Okay. The second study relates more to a more recent development where we have used metaproteomics to also use it as a tool to assess community composition. So one of the most basic questions that we always ask when we study any kind of microbiota or microbial community is, first thing we need to know is what is actual composition, who is there and how many of them, right? So when we have one of these microbial communities, and here's just a very schematic picture of a microbial community, we can assess its composition both by just counting the different numbers of the different species, and we would get cell numbers, but of course we could also assess the community composition by looking at how much mass does each member contribute, and what could that potentially tell us about how much activity everybody is contributing. Right, and so just to drive home that point, it actually could tell us very, very different things because microbes have a huge range of sizes and masses, right? And for this, just to, I have two pictures here. They are at the same scale, and it just illustrates in different microbial communities, microbes really, we have these tiny, tiny dots here, and we have really large ones. So there's up to like 10,000-fold differences in terms of mass a microbe actually can contribute if you think about a eukaryotic cell versus some kind of very small bacterial cell. Anyway, so at the time when we started with this study, there were a ton of different methods already available to do some kind of counting in your microbial community, right? You could, do sequencing-based approaches where you sequence specific marker genes, or use fluorescent hybridization, microscopy to really count cells. But there was really no validated method to actually look at microbial community composition in terms of mass contributions. And so that's where we thought metaproteomics could be a useful tool, because so that was really our goal, using metaproteomics to assess community composition in terms of mass contributions. And so the nice thing about microbes is that most of the dry weight mass is actually protein for almost all microbial cells. There's a few exceptions where we have specific storage compounds where that's not quite true, but generally most of the mass in a microbe is protein, so it can be used as a good estimator of mass contribution. So what we did in this case, and I was very lucky, I had a very talented undergrad student actually doing this work. What we did in this case now, we actually used these fully defined microbial communities that we had generated with different types of compositions to start developing metaproteomics into a tool that would allow us to actually get accurate mass readings on microbial communities. So we made these communities, we characterized them with metaproteomics, we did metagenomics on them, we actually also did amplicon sequencing on them, and then we spent a lot of time figuring out how we have to run these samples to really get good estimates. So it was a long methods development. Ultimately, and this is just one of the data points, it worked out pretty well. So here I'm just showing some data of one of these particular fully defined communities, which is a community where we had equal cell numbers in the community of each of the microbes. And so here are the theoretical compositions of these communities. Right here, Here we have what the community would look like based on just protein amounts that we put into the community. And here we have the community composition based on the cell number inputs, right? And as you can see, all the cell numbers were the same, except for this one where we had multiple strains that were so closely related we couldn't distinguish them. So that was what the communities looked like. And then we did metaproteomics or metagenomics to assess community structure. And as you can see, the metaproteomes are a relatively good representation of the mass contributions in the community, whereas the metagenome is actually a relatively good indicator of the cell numbers in the community. So once we had developed and validated the method, just to give you one quick example, actually I have two quick examples on how we used, how we can apply this now in different types of systems. So for example, here we studied phototrophic biomats from a soda lake in British Columbia. And so we went and, there's these phototrophic biomats and we just wanted to know their composition. So first thing we did is we did metagenomics and amplicon sequencing, and then we looked at community composition in terms of abundances of different taxa. And based on this sequencing-based data, you would say, well, the brown thing is the most abundant taxon, most relevant. And these are the proteobacteria. Whereas if you actually look at mass contributions using metaproteomics, the picture is very different. Actually, the green thing, which are the cyanobacteria, which makes more sense for phototrophic community, are the main mass contributors in this particular community. So with the different methods, we can get very different pictures of our particular system. And then just some more recent work that, I thought it was quite amazing. We currently, as I mentioned, we have a collaboration with Omri Finkel and Jeff Dengel at UNC, and my postdoc Fernanda Salvato is working on this particular systems where we have an Arabidopsis system that we can grow with a fully defined community. And in this particular case, we grew Arabidopsis with 4 different microbes, and we're doing this right now to optimize metaproteomics for root-associated microbiota. And one of the first things that we saw in the data is that actually there's a surprising amount of mass contribution of the microbes in this community. So when we just look at how many spectra we get from the plant versus from the bacterial members within the root, we see that up to 30% of all the spectra in the root are actually microbial. So we were quite surprised to see that, because we wouldn't have thought that microbes contribute quite that much biomass in a root. And of course, this is only detecting the proteinaceous part of the mass. And of course, the root has a lot of woody parts, so maybe, not the total mass. But in terms of protein, actually, the microbes are quite a large contributor within in the roots. Okay, good. So much about the biomass. So the last study, before going into a little bit of our more current work, the last study that I wanted to briefly highlight now looks a little more at yet another step further, where we use the metaproteomic data and the high-resolution mass spec data that we have to start looking at isotopes within microbial communities. And it's going to briefly get, I think, a little more technical, but I hope I can share some of the excitement we have for these particular approaches. So one of the goals here was to really try to see if we can extract any kind of natural isotope composition data in microbial communities, and I'll explain a little now why that might be relevant. Okay, so yeah, here I actually have a little bit of an example why you would actually care about learning the natural isotope compositions of these microbes. So let's, for example, assume that you have a microbial community that you have characterized really well using amplicon sequencing, shotgun metagenomics, even metaproteomics. So you have learned a lot about the composition and the potential of this community, and you have found maybe by comparing different conditions or something that one of these microbes in this community is particularly important. It's like a keystone species, and you have reconstructed its metabolism. So you have learned a lot about it. But then you run into like a little bit of an issue, which is, you find that one of the transporters that this microbe has is really abundant, and clearly has a major role in the physiology of this particular microbe. And if you have dealt with annotations of these types of transporters before, you know that usually you get to the level where you can say it's a transporter, maybe it's for a sugar, but that's usually where it stops, and you cannot, based on the sequence alone, infer more about its exact function. So you're stuck with the fact that maybe in this community you actually have different types of sugars present, maybe even the same sugar from different sources, and you don't really know which of these sugars is actually the driving substrate for this microbe. So wouldn't it be nice to have some kind of method that allows us to directly link this substrate to this microbe? And so that's what we're trying to do here now. Okay. So, and what we're using for this is a stable isotope. So as you know, in nature there's many most elements occur in multiple isotopic forms, and a lot of the isotopes are actually stable. And for carbon, that is true in that we have carbon-12, which is the most dominant form of carbon, is roughly 99% of all carbon. And then we have carbon-13, which is 1 mass unit heavier, and is roughly 1% of all natural carbon that is around. So, and when we look actually at a natural material and its isotopic composition, we usually compare it to a standard material. And so we compare it to the standard material, and if we find more carbon-13 in the material that we're looking at than in the standard, we call it heavier, and we give it a positive per mil notation. And if it has less carbon-13 than the standard, we call it lighter, and it has a negative per mil notation. So that's just some of the notations we'll be dealing with. And so the interesting thing is that a lot of natural materials and substrates actually have slight variations in their natural carbon isotope composition, right? So for example, if you look at different types of plants, C4 plants versus C3 plants, you see that they actually have different carbon isotope ratios, and we could potentially exploit this to look at who is eating what, kind of. And actually people have been doing that a lot already in the past. For example, in human studies, anthropologists often use this to link people to what they have been eating. This is just a little fun study that I'm giving here as an example, where the US Geological Survey, they took little fingernail clippings from all their visitors during an open house day, and they measured all the isotope ratios of the visitors and then tried to infer the different types of diets they may have eaten based on the isotope ratios. But I thought it was quite fun, but even humans even differ quite a little bit in the isotope ratios depending on what kind of a diet they eat. Okay, so using these isotope ratios has already actually been used quite a bit in biology before to infer different things. So there's two main questions that we can actually address with natural carbon isotope ratios in biology. And I'm just going to give you two brief examples before I actually start talking about the actual method that we developed. So the first question that is often addressed with stable isotope fingerprinting, looking at carbon isotope ratios, is what is the actual food source of a microorganism or of any organism? And so this is based on the paradigm that you are what you eat. An organism should have the same carbon isotopic signature as its food source, which is true in almost all cases where we have heterotrophic organisms eating organic stuff. And so, for example, just to give you one famous example for this, so we can look at this anaerobic oxidation of methane example. So what happens in anaerobic oxidation of methane is we have marine sediments where isotopically very light methane is produced in these sediments, which is going up in the sediments, and then it's actually oxidized by some kind of microorganism at a so-called sulfate methane transition zone. And so this is a really, really critical process that actually prevents all this methane from coming out into the water column and ultimately impacting the climate. So researchers have been very, very interested in what microbes are actually responsible for consuming that methane there. And so what they had found is that there's actually consortia of bacteria and archaea present at these transition zones, but they were never quite sure who is actually the consumer of the methane. So what they did in this case, they used isotope ratio mass spectrometry, where they had an imaging method where they could run across this such a consortium and measure isotope composition along the consortium. And what they found is when they got to the middle of the consortium, the isotope isotopic ratio was very, very low, which corresponded to the isotopically very light methane. And so they ultimately concluded that the archaea, which are in the center, are actually the methanotrophs. So here, this is a really critical example where they used the isotope ratios to figure out who ate the methane. Another thing where we can use this stable isotope fingerprinting is we can actually, for some organisms that actually fix carbon dioxide or carbon-1 compounds, they actually alter the signature of the carbon dioxide when they incorporate it. And they can use different fixation pathways to do so, right? You know, most of the plants would have the Calvin cycle, but there's also a lot of different other microbial pathways for carbon fixation. And each of these carbon fixation pathways actually has a different discrimination factor on how they change the isotopic signature of the carbon dioxide when they incorporate it. So basically with this we could also predict which carbon fixation pathway was used by a microbe. Okay, so ultimately it would be really nice to be able to measure these carbon isotope ratios for all the microbes in a microbial community. And it would be very informative if we could really learn what they might have potentially consumed in terms of substrate. The problem so far was there was no method to do that, right? So far, the gold standard method to measure these isotope ratios is to use an isotope ratio mass spectrometer. And what happens in this particular instrument, you take your sample, you burn it up, the whole sample as a bulk, and then you get like one value, which can be very informative, but you of course don't know all the subtle differences that might be going on in a microbial community. So that's what we wanted to resolve in this case. And so why we thought we would be able to resolve this is because we have high resolution data in metaproteomics, high resolution mass data in metaproteomics. And so here I'm going to give you one example of one of the hundreds of thousands of spectra that we acquire in one of these experiments. Right, so here is one of these spectra. Each of the peaks would correspond to a mass peak for one of the peptides. And then, of course, we can identify these peptides, assign them to an organism. We know its sequence. We know a lot ultimately about each of these peaks, right? We know the sequence. We know the organism of origin. And actually, when we zoom in one of these peaks now, what we see, of course, is that we have, for each of the peptides, we have multiple peaks because we have isotope forms, right? The first peak of the peptide would, of course, be the peak where all the atoms in the peptide are all of the most dominant isotopic form. And then in the second peak, one of the hundreds of carbon atoms, for example, would be exchanged for the heavier version. So we get a whole series of isotopic peaks for each peptide. And so our thinking was that, if we just quantify these intensities of these peaks and did some kind of complicated math, we should be able to get at the isotope ratio of each peptide and ultimately at the isotope ratios of the organisms where these peptides came from. So with that in mind, I was very fortunate to work with two very talented bioinformaticians, Mark Strauss and Xiaoli Dong, where we developed this particular tool. So we developed ultimately an algorithm that actually allows us to do quite that. So what we do now in this algorithm, just in very rough terms, is we actually simulate in silico all the different isotope patterns for every given peptide, hundreds of different patterns that would be possible for each given peptide. So we simulate peptide isotope patterns, different intensities for the different peaks, and then we compare them to the actual experimental data, and then we do a comparison, we do a goodness of fit analysis, and ultimately we say, well, this peptide must have this isotopic value based on this comparison. And that's not a very accurate method, but if we do it for enough peptides for each organism, then we can actually get a fairly good reading. And so after developing the method, along the way we did a lot of proof of principle benchmarking type of studies using pure culture organisms where we use different bacteria and archaea. We measured them. Having pure cultures allowed us to actually measure them in a gold standard method, which is the isotope ratio mass spectrometer, and we compared it to our newly developed protein stable isotope fingerprinting method. And as you can see, we get a relatively good correlation of the values, so we can get relatively good readings on that. And we also found that some of the autotrophic organisms that have a lighter signature, the heterotrophs have the heavier signatures, all the things that we expected. We also, at that point, we found that we were using BSA as our quality control in the mass spectrometer, and we actually found that this cow most likely must have eaten a lot of corn that provided this particular BSA. And actually, part of the method is that we have to use a calibration standard to get rid of some offsets that we have in the instrument, and we actually ended up using human hair because it has it mostly consists of protein, so it's a really nice sample where we can fully quantify the isotope ratio and actually measure it in a mass spec. So that's our calibration standard, and it falls right where it's supposed to go. Okay, so Now I'm going to give you two little examples of how we now have applied this method to address both the types of questions that I introduced earlier. So the first one is going back to the worm and back to the question, who has eaten what, or what was the actual substrate source, right? So we already introduced the worm that has the symbiotic community, and we use this as a case study because we already knew a lot about what's going on in this particular worm, right? So we had a really good model about where are the carbon sources for each of the members of this community, or at least we thought so, right? So based on all the metagenomic and metaproteomic work, what we thought we knew is that we have two primary carbon fixation events, all the carbon in the symbiosis coming in through the carbon fixation by these two symbionts, the carbon then later being handed off to the host and ultimately also being used up by these sulfate-reducing symbionts. And we had actually one symbiont where we didn't quite know yet what it was doing. So that was our idea of the carbon flow. So ultimately all the carbon comes from these two primary fixation events. So when we measure a whole worm, what we also knew is that when we get a bulk value, it's around -30. So that's the two things. So based on these two things, we hypothesized that all the values in this particular symbiosis should be somewhere around this value because all the carbon is derived from the same primary carbon fixation events. So somewhere between -25 and -35, accounting for a little bit of an error in our method. So we used our method to measure it, and what we found, fortunately, most of our predictions were right. So most of the symbionts are somewhere in the realm of -30, -34, -31, -30. We actually learned a lot more about this particular symbiont. The host is also still within the range that we had predicted. But we also interestingly found that some of our predictions are not right because we had one symbiont that has a signature of roughly -18, which is very far outside of the expected range. So clearly this particular symbiont must have a different source of carbon that is not a source from within the symbiosis. And by now we have some vague ideas about it, most likely being some polyphenolic compounds potentially that come from a seagrass that the worm associates with. Okay, so that was relating to the first question, learning about substrates. Here we really could follow the substrate and see if the substrate is linked with the specific microbes. The other type of question I had introduced earlier is that we can also learn about carbon fixation pathways. And for that I'm going to talk very briefly about a study that Brandon Seher did with us. He was a PhD student at the Max Planck Institute in Bremen. And so he was looking actually at a marine ciliate. So this is a single ciliate cell, a small, very tiny protist that lives in marine sediments. And the white stuff that you can see, like this furry coat, is actually bacterial symbionts that live on the outside of the ciliate. Roughly 1 million cells per ciliate. So it's very, very little material, so we had to deal with that. It's roughly around 10 micrograms of wet weight material that we could get from these particular ciliates. So when Brandon started studying this system in his PhD, his assumption was that these symbionts that sit on the outside of the ciliate must be autotrophic organisms that fix carbon dioxide to ultimately provide some nutrition to the ciliate that maybe just ingest those symbionts ultimately. So he sequenced a lot of metagenomes from a lot of different ciliate species, and he scrutinized them for any evidence for any kind of carbon fixation pathway in the genome of these microbes to figure out how do they actually fix the carbon dioxide. And he was very frustrated for quite a long time. I think he spent more than 2 years on trying to figure out what was wrong with his metagenomes because he couldn't find any of the known carbon fixation pathways in any of the genomes. So he did a lot of resequencing and reassembling and all kinds of methods to get better metagenomes. And ultimately he concluded maybe they're actually not autotrophs, right? So it was which was his primary assumption initially. So ultimately he said, most likely there must be heterotrophs or mixotrophs, but they're not able to likely to fix carbon dioxide in major concentrations. So we at that point came into the project and helped him to actually analyze the isotopic ratios of the particular microbe. So we measured the isotopic ratios of the Kentrophora symbionts, and as you can see, their signature is relatively heavy, right, above -10. And then, whereas we would, if we had any kind of an autotrophic carbon fixation pathway, we would actually expect them to be in this particular range. And then, so Brandon went through a lot of work to actually figure out in all the literature what other microbial symbiosis with autotrophic and non-autotrophic bacteria, what kind of signatures they have. And, of course, he found it for a lot of the systems where we have these autotrophic symbionts, we have a very negative signature, and none of them corresponded to this signature. So here in this case, the Protein-SIF method allowed us to provide more evidence that these particular symbionts in this particular habitat are not autotrophs. Right, so actually the opposite of what he was looking for initially. Okay, so just a quick outlook on where we are taking this particular method right now. So Angie Mordan, my student, is actually today with me. Here she's now working on a project where we want to use this stable isotope fingerprinting method to actually link the species in the microbiota directly to the different dietary components that they're linked to. And currently we know very, very little about it, right? There's a lot of evidence or indirect evidence where we see when people or mice eat a specific diet that is enriched in a certain compound, we see correlations where microbes go up or down, and then we assume that maybe they are feeding on the specific dietary component. But of course, it could also be all kinds of cross-feedings or secondary effects. And there was up to now really no way to directly test this. So we are now using these isotope-based methods to actually start linking specific microbes to the dietary components that they actually live on. So that's one of the current projects we have in the lab. Good. And I know I don't have much time left, but it's just a few slides about a current study that we're now doing also in the realm of diet microbiota. And this one really looks at the fate of dietary protein and what it does to the microbiota and the host. Okay. And I think I have my other student, Alexandria Bartlett, who is also here today on one of the slides. She's actually leading that particular study. Okay. So this particular study relates to the fact that we have been learning over the last decades a lot about the diet really impacts the microbiota and ultimately, of course, has also a lot of health impacts on the host. And then, of course, there's feedback effects and all kinds of interactions going on. And just to give you one of the many, many studies that have been done up to this date is there's this really beautiful study that comes from the Eric Martens lab where they looked at feeding mice a diet rich in fiber and a diet with completely depleted in fiber. And what they found is that when the mice are feeding on a fiber-free diet, the microbiota actually starts attacking the mucosal layer and degrades it, and ultimately that can lead to pathogen invasion. Right? So really serious effects that the diet has on the microbiota, ultimately leading to serious host health outcomes. And so there have been a lot of studies, and I'm sure many of you in the room are aware of that, looking especially at fiber and fats in diets. And there's been a lot, we have learned a lot, but I think there's still a lot to learn in this particular realm. For us, of course, working in the realm of proteins, we were wondering about what happens in terms of proteins, right? How does dietary protein affect the microbiota? And what we found is, when Ali actually did a very large literature survey, there's very, very little known right now. There's almost nothing in the literature that really relates to the interactions of microbiota with dietary protein. The little that we know comes from different types of studies. For example, there is the Nurses' Health study with more than 130,000 participants that, for example, found that when replacing animal protein with plant protein, that was associated with a lower overall mortality. So it's an interesting finding. Then there is other studies, although the evidence is still quite weak, where they have looked actually at different types of dietary protein sources where they have observed certain microbes changing up or down in abundance in response to different dietary protein sources. And there is actually quite a lot of literature more looking at on how protein can actually have health impacts on the hosts. And one of the main mechanisms is protein fermentation, which occurs when protein actually gets to the colon and gets fermented by microbes. And that can actually produce a lot of potentially toxic metabolites. It can lead to all kinds of detrimental health outcomes such as colorectal cancer or ulcerative colitis. So there's quite a lot of things that are potentially going on with protein, but we know very, very little about it. And so, of course, now with the metaproteomics approaches that we have developed, we have now a tool in hand that allows us to look at everything that is going on with protein in the intestinal tract, right? We can look at the host proteins in the intestinal tract, we can look at the dietary proteins in the intestinal tract, and we can look at the microbial responses in their proteomes in the intestinal tract as well. So, ultimately we're going to use similar approaches to what I have introduced already earlier in this particular study. And one of the first experiments that we are now doing is based on the hypothesis that some dietary protein sources have a larger proportion of proteins that are actually resilient to host digestion, and that would ultimately have more impact on the colonic microbiota. And there's a little bit of literature that talks about that there's actually different types of proteins and different types of dietary protein sources that differentially impact digestion in the upper intestinal tract that might make it better to the lower intestinal tract. So in this one of these first experiments that we actually have just now carried out, we actually look at conventional mice and mice that are germ-free, so we can actually differentiate the microbiota effect on diets. We have developed fully defined diets for these mice where we have a single dietary protein source in each of the diets and everything else stays the same in these diets. And we have different types of protein sources: casein, egg, yeast, rice, soy, all kinds of different sources. And we feed these mice groups these fully defined diets now, and we start collecting, we have collected their stool samples on different days throughout. For each oscillation is 7 days long, and so we're oscillating all these diets. Ultimately, we sacrifice the mice after feeding on either a soy-based diet or casein-based diet, and we also collected different parts of the intestinal tract throughout in the end. And so now we really wanted to determine the fate of the dietary proteins from the different sources. So actually, how does the dietary protein change throughout the intestinal tract? Is there actually any differential effects? And ultimately also to look at how do different dietary protein sources impact the microbiota. And so we have only very few preliminary data so far, but I thought it was, there's some quite interesting things, just looking again at mass distributions, right? If you have if we feed a diet that has actually wheat as one of the main protein sources, we look at the germ-free mice, we identify mostly mouse proteins and wheat proteins. So that's nice to see. But if you actually look at a conventional mouse, most of the protein that we measure in terms of mass is actually bacterial derived. And we are not completely firm on these numbers. These might still change quite a bit, but ultimately it's impressive to see that actually most of the mass in terms of protein in the colon is ultimately the bacterial protein. Everything has been converted to that. Another first result from this particular study, again, we had mice that were fed with wheat proteins, with a wheat-based protein diet. We looked at how do the dietary proteins in the stool of these mice differ between germ-free mice and mice with a microbiota, right? And so here we are looking only at the proteins that are the most fold increased in germ-free mice, right? So there's a lot of proteins that are much, much more abundant in the stool of germ-free mice as compared to the conventional mice, where the microbiota also has an impact on the dietary proteins. And just to zoom in on some of those major proteins that are most different between the germ-free mice and the microbiota-containing mice, we see a lot of these proteins that initially at least are annotated as inhibitors of digestive enzymes. So clearly there seems to be something going on where we have an enrichment of proteins that are inhibitory to digestive enzymes, and that could be a good explanation. Ultimately, the microbiota then seems to efficiently remove those later, but of course they provide a substrate for the microbes. And now we're taking it from there and going to see what happens next. Right now everything is on a mass spec running right now, so we're really happy that that's going on. So I'm coming to the end of my talk. Just to sum up, I hope I was able to show you today that there's really a lot of different questions that we can address using metaproteomics, right? We can look at abundances, we can look at physiology and metabolism, we can learn about interactions of microbes and their hosts, and we can learn now even about different carbon substrates used by these microbes. And currently we're actually also developing the tools more to also use artificially labeled substrates that we can feed in different types of settings. If you're interested in learning a little more about metaproteomics, this year I actually wrote a perspective article. It's a very short perspective article, but I tried to give a brief overview of all the main questions that you can address with metaproteomics and some of the more central references in the field. Okay, and with that, I would like to thank all the amazing people that are involved. I already highlighted some of the people in my team that are involved in this research. We have some really good collaborators at NC State that work in the realm of mice and bioinformatics, Ben Callahan and Casey Terriot, and then a lot of people at different institutions, and of course our funding sources.